just want to remind you that these seminars happen every Tuesday, and we are bringing together scientists who are interested in the interface between the nervous system, organ physiology, immunology, aging, cancer. So stay tuned for any updates in our website and also in Worldwide Neuron seminars. Before introducing our wonderful speaker, I also want to remind you that you can ask any question during this talk using the Ask a Question button here. And also, if you want to meet the speaker after the talk during a Zoom Q&A session, I will write the Zoom link in the chat in a bit. So now for real, I am eager to introduce Dr. Robert Wessells, and I am going to briefly summarize his career so we have time for his talk. Dr. Wessells is a professor at the School of Medicine at the Wayne State University. He graduated with a bachelor's in zoology from Miami University before acquiring a PhD in molecular genetics from Ohio State University. He received postdoctoral training in fruit fly genetics and physiology at the University of Michigan and the Burnham Institute for Biomedical Research before taking his present position in 2014. His research focuses on understanding the molecular genetic mechanisms that underlie the benefits of exercise, and he uses the Drosophila model system to understand either neuronal factors that control exercise behavior, muscular factors that control adaptation to training, and also adipose factors that regulate metabolism in response to training. So today he is going to be talking about brain muscle signaling, coordinating exercise adaptations. Thank you so much, Robert, for being here with us today. It is a huge pleasure, and the stage is all yours.
Okay, thank you so much for the invitation and the very nice introduction. I'm happy to be here and to talk to you about some of the work my lab has been doing for the last 5 years in how the brain and muscle work together to coordinate adaptations to chronic exercise. So why are we interested in this? We're interested because endurance exercise is so far the best intervention that has been discovered for promoting healthy aging. And by healthy aging, I mean both retaining more youthful and healthy function, like you see with these advanced aged athletes, and also avoiding age-related diseases. Chronic exercise is known to reduce the incidence of just about every age-related disease, including most of the world's major killers, such as heart disease, cancer, Alzheimer's, diabetes, etc. So for us, we're very interested in understanding what the molecular mechanisms are that drive these adaptations in chronically exercising animals, and how we could leverage those as therapies to lower the incidence of these types of diseases, perhaps even in people who can't exercise. So to do that, we wanted to bring to bear some of the powerful genetics of the Drosophila system. So we figured out a way to exercise flies using this device that we call the Power Tower, which is it's named after an amusement park ride near where we are in Detroit. And this machine raises and drops a platform full of vials of flies, and it takes advantage of their innate instinct to climb to the top of the vial once they get dropped. So even those of you who may have never worked with fruit flies, you've probably seen or at least heard neighbors working with fruit flies tapping them down all the time. And when you tap them down, they go straight up to the top. And what we found was that they'll continue to do this over and over. So you can use this machine basically in effect as a fly treadmill, because once they get to the top, we just keep knocking them down and they keep running. And if we do this in a defined paradigm of gradually increasing amounts of daily exercise, what we find is that trained flies are able to run much faster up the side of the vial than genetically identical age-matched controls, which are only different in the fact that they have not had this chronic exercise. And so this little video is going to show you how that works. Oops, I may have to get out of the pointer option first. Right. No, not that text bar. Uh-oh. Just 1 second. I think I need to turn off the pointer in order to show this video. Okay, here we go. Okay, so as you can see, this is a roller arm. It's not a very high-tech device. It has a skateboard wheel here, which pushes down this lever, which raises the platform and drops the flies. Now, as it zooms in here in a second, you guys can all notice when the flies are dropped, notice how they'll all start running up towards the top. Okay, so this is running straight up the side of a wall. Turns out to be pretty good exercise. And I also want to point out that these are the unexercised controls. We keep these on the machine exactly the same way and receiving all of the same non-specific stresses that are associated with the machine. The only difference is that we push the lid all the way down so that they really can't run up the side or get any of the exercise. And so this allows us to attribute the differences between the unexercised and exercised groups to the actual running, not to anything else that happens to them on this machine. And there are a lot of changes that happen. As you can see, comparing exercise to genetically identical age-matched controls, the speed that they're able to climb up the vial increases. It still goes down with age, but across ages, they remain faster than their unexercised cohort. We also see an increase in the amount of time they're able to run. And I'm going to be using this run span diagram a lot during the course of this talk. And what this is are each of these dots represents a vial, and we note the time at which each vial stops running and then graph them as a curve going downwards. So that's why we call it a run span, because it's similar to a lifespan curve. So not only speed and endurance, but also even aspects of performance that aren't directly related to the training, such as flight, as you see going up here, and the resistance to pacing-induced heart failure is also increasing in exercised guys. The fractional shortening in the unpaced heart goes up, and we also see several lines of evidence that point to increased mitochondrial number, including increased activity of mitochondrial enzymes such as aconitase. I'm going to bring the laser pointer back. And we also use this particular assay as a biomarker for the, quote, exercised state that these animals get in, because we see a dramatic increase in lysosomal activity as evidenced by lysotracker puncta shown here. Now, this is reflective of all kinds of lysosomal activity. And in terms of exercise, we think it's mostly lipolysis and autophagy that we see reflected here. And this is a really reliable marker for us to see whether animals are in an exercised state. Okay. So these are the basic things that we observe in wild-type exercisers. So how did we get interested in the brain? Well, it started because we wanted to address this question of why in the world the exercise response in Drosophila is sexually dimorphic. So as you can see here, males and females have a pretty similar baseline climbing speed. But if you put them on the exercise machine, for some reason, the males improve their speed, as I just showed you, but females actually stay pretty much the same. And this is also true of this lysotracker biomarker of increased lysosomal activity. You see exercised males get this huge, almost 30-fold increase in lysotracker puncta, but females who undergo the same exact exercise training don't do this. And this is not because of any lack of inherent ability to complete the exercise program, because as you can see here, females at baseline, they have a somewhat lower endurance than males, but they're still able to run 200 minutes, which is, much longer than the actual daily exercise time. So there's no obvious reason why females should not be responding the way that males do. So we set out to figure out why, and we thought maybe we could use females as a really serendipitous kind of model for animals that have a poor training response, because there are, as I'm sure you're aware, a great deal of variation in animals of all species in terms of how well they respond to the same training program, even in humans. Many of you may have noticed that some of your friends respond better to exercise training than you do. So we thought this might be a good way to get at how some of that works. And we took advantage of the fact that in flies, sex determination is in fact tissue autonomous, meaning that we can change the sex of specific cells or specific tissue types, and we can do that after they grow completely to adulthood, so that we're only changing gene expression and not changing the actual anatomical differences between male and female. And it turns out that one particular RNA splicing factor called transformer is responsible for driving male or female specific gene expression in the fly cell. So what we did was use transformer, put it into various tissue types, and see which one, when we feminized that particular tissue, would make the males stop responding to exercise, with the logic that that must be the tissue that is causing the difference between males and females. And we thought that by doing this, maybe we could identify some factors that could confer these exercise benefits by finding out what was lacking in these females. Okay, so I'm going to skip all the negative data. We found that actually feminizing muscle made no difference. Feminizing adipose tissue made no difference. But when we went to feminize neurons, if you take a look at these males, the RU minus males are the ones that did not receive the drug that induces the trans gene. So you see they're normal here in gray and black. The ones that exercise are faster than the ones that did not exercise. But the ones that received the inducing drug and expressed the feminizing transformer construct specifically throughout the nervous system, those are here in the two different colors of blue. And you can see that they got no benefit from exercise at all. And this was also true in terms of endurance as well and other assays. So that told us that the main difference between males and females in responding to exercise is centered in the nervous system. So we wanted to know what part of the nervous system is the important part. So we did a sort of targeted mini screen here where we used drivers that mark different subsets of neurons that we hypothesized could be important, and then tried them all to see if feminizing just these subsets would render males no longer able to respond to exercise. And in every case, that did not work except for this one subset, which is marked by the enzyme TDC2. Now, TDC2 is an enzyme that produces octopamine from tyramine, so this driver marks specifically neurons that produce octopamine. And this subset of neurons by itself, whether it is female or male, can make the whole animal react as if it is female or male. So it's clear that this is very important to the exercise response. So does it make sense that octopamine would be a big part of the exercise response? Well, it sort of does. Octopamine is the invertebrate equivalent of norepinephrine, which is widely known to be important acutely during a bout of exercise to increase heart rate and energy uptake in muscle. It does the same thing in flies. But you can see that it also might make sense that it's involved in these sort of chronic adaptations as well, because it's involved in a lot of different behaviors, including some behaviors that are actually already known to be sexually dimorphic, such as aggression and obviously courtship, and also some behaviors that are not known to be particularly dimorphic, like learning. So it sort of made sense that maybe octopamine is causing this difference, and we hypothesized that during a bout of exercise, somehow males are releasing octopamine into the circulation and females are not. So to test that hypothesis, we thought that perhaps we could make females respond better to chronic exercise if we simply fed them octopamine, and that turned out to be the case. So if we feed females octopamine, mix it into their food during the 3 weeks of their exercise training program, you see the normal ones without octopamine fail to respond, just as I showed you before. They get no benefit to endurance or speed, as you see here in the gray. But the ones that were fed octopamine now get this huge benefit from exercise. And what surprised us and was especially exciting was that even the unexercised ones now look as if they've been exercised. And furthermore, the ones that have octopamine and are exercised are getting no further benefit, which is consistent with the idea that exercise is actually working through octopamine. They're not working in parallel. And we see that this also works on unexercised males. So the males that are not fed octopamine they get a benefit from exercise. If you feed them octopamine, they now get a benefit whether they're exercised or not. So this works in both males and females, and it works in terms of endurance, it works in terms of speed, it works in terms of flight. However, we're feeding them pretty large amounts of octopamine, so this may not be physiological. We wanted to try something where we felt confident that they're actually having a normal physiological amount of octopamine that they would get if they were actually exercising. So what we did is we put a temperature-sensitive depolarizing construct specifically only into octopaminergic neurons. And once we had that construct in there, we put them at the inducing temperature. We put all the controls also at the same inducing temperature. And we asked if that allowed females now to benefit from exercise. And as you can see, once again, none of the controls in the females benefit from exercise, but the ones that have these neurons depolarized not only benefit from exercise, as you see in the dotted line, but they also are out here with the same amount of endurance, even when they're unexercised. And you see the same thing over here in males, with the only exception being that the wild type ones with no depolarizing also benefit from exercise when they're male. But unexercised males also benefit just from having these neurons depolarized. In other words, just from having octopamine released into the circulation, they're able to get the same benefits to speed and endurance that they would get from a whole 3 weeks of exercise, which to us seemed pretty extraordinary. But again, we thought, well, maybe this is a little bit over the top because we're depolarizing these neurons throughout the whole 3 weeks. And we know that in real life, whether you're a fly, a mouse, or a human, you're really not exercising 24 hours a day. We want to know if this would work if we depolarize the neurons only during the time that they were exercising. And importantly, this also makes it more translational, because in an animal with a closed circulatory system like a mammal, having norepinephrine flowing through all the time would have devastating effects to the cardiovascular system. So that's obviously not a therapeutic option. But pulsatile expression of these, as you would get from simply doing, let's say, half an hour of exercise a day, might actually work. So we tried that. We tried intermittently depolarizing these neurons by keeping them at the uninducing temperature, except for all the minutes of a normal exercise program. And we took side-by-side wild types that we put into the normal exercise. We took ones that were depolarizing their neurons and just depolarized their neurons without actually running. And we took ones where we depolarized neurons and ran them all at the same time. And what we found, this is just to show you that this didn't affect their baseline endurance, but what we found after the 3-week program was that animals that depolarized these neurons, whether male or female, whether exercised or not, got a huge boost to endurance. They got the same boost to speed, the same boost to cardiac health, the same boost to lysosomal activity. All the characteristics that we have noted in wild type exercise, they got just from depolarizing these neurons for about an hour and a half to 2 hours a day. So this was really exciting to us. And this summarizes sort of this first series of experiments. We know now that if we put animals on a treadmill, they're going to activate octopaminergic neurons, which is going to release octopamine into the circulation and is going to have phenotypic effects on skeletal muscle, on heart, and on adipose tissue. And crucially, and perhaps surprisingly to us, we found that even if we take a couch potato fly and simply activate their octopaminergic neurons by a genetic manipulation, we get exactly the same effects, even if the animal is never running at all. And in fact, we even got it if we restrained them so that they're actually having lower activity than wild types. We still got all of the benefits of exercise, provided we activated these neurons, and we could get the same result by simply feeding them octopamine as well. So it's clear that the effect of these neurons is also dependent on the release of octopamine. So the next thing we wondered was, what does octopamine do once it gets into the circulation? How is it received by these different tissues? And what receptors are necessary to receive that signal? So we did a project where we took what was at the time the 4 known octopamine receptors, There's another one that's been discovered since we started this project. But we looked at these four: OAMB, which is an alpha adrenergic receptor, and these three are beta adrenergic receptors. And what we did was just knock them out in every permutation of these different tissues and ask which one of these knockouts blocked exercise from inducing these improvements. And what we found is summarized. I'm not going to go through all the data in this paper. I'm just going to show you the summary here. And we found several interesting things. So there are examples of one particular receptor, such as beta-3 here in the fat body, which is by itself required for the fat body to induce lipolysis. And none of the other receptors are required in this tissue. We also found, however, examples where there were multiple receptors which were required in the same tissue. And this was a little bit surprising to us because most of these receptors, as far as we know, act through the same basic downstream components involving either cyclic AMP or PKA signaling or PKC and calcium modulated signaling. It's not really obvious why these receptors would not be redundant, but they are not. So we're currently investigating what is different in the downstream effects of these receptors. And another factor we discovered that was pretty interesting to us was that in several cases we found that knocking out a specific receptor in one tissue actually blocked the effects of exercise in another tissue, which strongly implies to us that downstream of these receptors, there are second messengers being activated, and these are likely to be myokines that are going to be secreted and communicate with other tissues, or adipokines, which are going to be coming from the fat and communicating with other tissues as well. And it's well known that exercise induces these kind of effects in mammals. So we're working now to try to figure out how this works in flies. And in a few minutes, I'm going to show you an example of a putative one of these myokines that we may have discovered. So the next thing that we wanted to do, now that we know that octopamine has to come out of the brain into the circulation, and we know that it has to be received by specific receptors in all these different tissues, we started focusing in on what happens after the signal is received in the muscle. And we wanted to find out what genes have to be upregulated in the muscle in order to execute the program of exercise adaptation. And one that we've done a lot of work on, together with our collaborators at University of Michigan, is cestrin. So cestrin is a stress-inducible protein. It was originally isolated as a target of p53 in terms of genotoxic stress. It was later discovered to be activated by FOXO to respond to hypoxia and also oxidative stress. And in the intervening years, it's been shown to be induced by many other stresses as well. Its enzymatic activity is to be an antioxidant, but its more important activity, it turns out, in exercise is not to directly reduce oxidative damage, but instead to inhibit mTOR signaling. It inhibits both TOR complex 1, and in contrast to rapamycin, it inhibits TOR complex 1, but it actually stimulates TOR complex 2. And we think this modulation, well, we know, in fact, that this modulation of metabolism is critical for exercise adaptation to take place. Exercise adaptations do not take place if you knock out cestrin in muscle. And if you overexpress cestrin in muscle, what you see is that endurance, as shown here, and also speed and all the other downstream effects of exercise adaptation can happen even in unexercised animals. And we found that an important downstream player, downstream of cestrin, is the Drosophila version of PGC1 alpha, which is a well-known exercise response gene in vertebrates. In flies, the PGC1 is referred to as spargle, so you'll see spargle in some of the upcoming slides. It's the same as the fly PGC1 alpha. And so we published a couple of papers on cestrin in exercise in recent years, and those are all available online. So I'm not going to go into any more details about this, but right now, instead, I'm going to move on to some unpublished data. But I do want to introduce to you cestrin and spargle as being very important in the exercise response in muscle, because I'm going to come back to those later in the talk when we start looking at some therapeutic tests. Okay. So downstream of Sestrin and Spargle, we think we've identified another new important exercise player, and I'm going to show you some of the recent results from this project, which we're in the process of publishing but are not published yet. So our collaborator on this project, Junhee Lee, discovered this previously uncharacterized gene in flies, which was important for regulating autophagy in the eye. And when he did blast searches on it, he found that it was actually homologous to a vertebrate gene called FNDC5, which we were pretty excited about. So we decided maybe this would have something to do with exercise-induced autophagy in the fly. So why did we think that? Well, because in the mouse, FNDC5 is an exercise-induced protein, which when it receives signals from either exercise or from cold, from the environment, is cleaved to produce this exercise, what is sometimes referred to as an exercise hormone called irisin. And Bruce Spiegelman's lab has worked on this a lot, along with some other labs, and they've been able to show that exercise strongly induces FNDC5 RNA and protein, and that it causes this shorter version to be cleaved and reduced into circulation. So it is a prominently studied putative exercise-induced myokine in vertebrates. And there is some controversy, however, to its actions and to some of its mechanistic implications in the vertebrate systems, because it's extremely difficult to detect, and its role in the circulation can be transitory. So there's always controversy about how much of it is in the circulation and where it's going. So we thought maybe the fly would be an interesting way to study this. And because it has been shown to be important to responses for exercise and cold, and in response to cold, it induces fat browning, which is a hot topic of study right now. Many of you are probably familiar. That's when white adipose tissue increases mitochondrial uncoupling and becomes more thermogenic and takes on a browner appearance. So one of the effects of both exercise and cold is to start white fat turning into beige fat, and thereby improving metabolic efficiency. And so graduate student in my lab, Tyler Cobb, decided to work on this project for his PhD. And we decided to, since it's involved in exercise and in cold, we would name it Iditarod, because that's associated with exercise and cold. So the fly name for this gene is now Iditarod. And we've been able to show that Iditarod is induced upon exercise, both at the transcript level and also at the protein level. We've also been able to show that, just as has been shown in mice, PGC1 alpha heavily activates Iditarod, just as it activates FNDC5 in vertebrates. And I want you to take note of this Iditarod rescue. So the mutant version of this in flies is actually caused by an insertion, and that insertion has placed into that region of the genome a driver element, which will now can drive transgenes in the expression pattern of Iditarod. So we created a fly here where we expressed Iditarod in its own expression pattern in this mutant background, in order to show that the phenotypes that we're seeing in this mutant are in fact due to this locus and not to some other locus. So when you see the Iditarod rescue in future slides, that's what that is. Okay, so the mutant Iditarod flies have dramatically impaired endurance, just like we saw before with Sestrin and Spargle. And if we put this rescue construct back in, the endurance comes back to normal. Now, in contrast to what I showed you with octopamine and to what I told you about Sestrin and Spargle, all of those impacted endurance and speed side by side, as if they were all operating as a suite together. But now we're getting, we think, far enough downstream where we're starting to see things that have stronger impacts on one aspect of exercise adaptations than the other. And this is one of those, because it has this very, very strong effect on endurance, but it has very little effect on climbing speed. And we know that muscle is the key place where Iditarod needs to be expressed in order to respond to exercise. because if we simply do an RNAi that's located only in muscle, we see very similar effects to the entire genomic mutant on endurance. We also find that Iditarod mutants can no longer adapt to exercise training. So if you look at these guys here, here's the wild type background getting a nice improvement to endurance with exercise. However, the Iditarod mutant doesn't get any better with exercise whatsoever. It's already lower at baseline and it can't get any higher. Now, again, this is specific to endurance because, as I showed you on the previous slide, speed is pretty unaffected. Also, lysotracker in the fat is pretty unaffected, which tells us that the animals themselves are experiencing the sensory input of exercise. They are doing portions of the exercise response, but without Iditarod in muscle, they still don't improve endurance. And we know that muscle is the important tissue, again, because a muscle-specific RNAi has pretty much the same effect as the genomic mutant. So that means it's required, but what about being sufficient? Because we're looking for things that could be therapeutic. We want to know if we put Iditarod into the muscle, will it make them look as if they've been exercised? And the answer is yes. Here you see if we simply express Iditarod only in muscle, an increase to endurance that is substantial. We also see if we look at exercise versus unexercised, in wild types, you see the nice increase in exercise. In Iditarod overexpressors, the unexercised already look like wild type exercised. And if you then exercise the overexpressors, they don't get any significant further benefit, which again is consistent with all of these things working through in the same pathway, not in parallel. And to show that even more definitively, in these experiments, we showed that Iditarod is upregulated by octopamine, showing that it is downstream of octopamine. And also in Iditarod mutants, octopamine is no longer able to increase endurance as it does in wild types, as I showed you at the beginning of the talk. So we think that all of these things are working together in the same pathway that we're starting to pull together from all these different projects. Exercise causes octopamine release, and in a muscle cell, we have octopamine receptors that pick up octopamine, activate downstream activities that cause upregulation of cestrin, and then upregulation of the PGC1 alpha homolog spargle. We have reason to believe, based on sequence homology of conserved NRF2 binding elements in the Iditarod promoter, that NRF2 may also be involved here in this process. We're currently examining that genetically. And this entire cascade results in upregulation of Iditarod transcript, and that leads us to many of the downstream effects of exercise. I put this X over autophagy, not because exercise doesn't increase autophagy. Exercise does increase autophagy, but we found that this is actually not required for the increase in speed and endurance. So that's what I'm referring to with this X. It may be required for other metabolic impacts of exercise, of course. What we don't know yet, which is a major question mark that we're working on right now, there is a consensus cleavage site in the fly Iditarod, which gives us a predicted small peptide. We are working on trying to detect that peptide using mass spec. We have not been able to do so as yet. And we're also working on trying to distinguish, as I mentioned earlier, what the different octopamine receptors are doing in between the receptor and upregulation of cestrin. Okay, so that's a summary of about 5 years of work looking at the wild type responses to endurance exercise and how those are translated into improved function. Now, for the last few minutes of the talk, I want to talk about some of our recent ideas for how we can put some of these molecules to work as therapies. And I'm going to just talk very briefly about two different things, one of which was we're going to try stimulating sensory input to the brain in order to mimic exercise, similar to how we did in flies by depolarizing octopaminergic neurons. And we're also going to try using some of the genes we've identified that are induced by exercise, in this case cestrin, in order to provide benefits of exercise to disease models. And in particular, we're interested in disease models that themselves affect mobility, such that a patient who has these diseases actually cannot exercise. So these involve neurodegenerative diseases and also mitochondrial diseases. Okay, but first I'm just going to show you a couple of slides about our ideas about using virtual reality to mimic exercise. So since we know that in the flies, if we bypass the sensory input from exercise and just depolarize octopaminergic neurons, that in itself can coordinate a response among multiple tissues. We have a hypothesis that maybe the same thing goes on in humans. So when a human starts exercising, it stimulates their brain to coordinate a response which is dependent on norepinephrine, which goes to muscles and heart, and that a pulsatile, short-lived daily amount of this could actually help to coordinate a healthier heart and skeletal muscle. And the way we're going to try to do this kind of pulsatile norepinephrine is not by any kind of pharmaceutical means, but by simply using virtual reality to provide sensory images that tell the person's brain that they are exercising. And we believe that it is not going to be required that they consciously believe that they are exercising. They'll know that they're using virtual reality, but we think that the autonomic response will generate norepinephrine release anyway, just as we've all experienced at, for instance, a roller coaster or a scary movie. We know that we're not actually in the movie being chased with an axe, but we still increase our heart rate, and that's because we're releasing norepinephrine. And our vision for this is that if we can test this sufficiently and prove that this can work, what we can do is apply this to bedridden patients in hospitals, because as many of you will be aware, patients go into a hospital for the purpose of an illness, for an injury, and they're on extended bed rest while they're recovering from that. Now, on top of whatever put them in the hospital in the first place, they're also now experiencing secondary complications that simply come from being sedentary. So we think this population, which is an enormous population around the world, could be susceptible to a really inexpensive, non-invasive therapy, which would be just using virtual reality exercise to give them sensory input of exercise, which would increase heart rate and coordinate various other metabolic responses to reduce the negative impact of being sedentary for the duration of their hospital stay. And so this is something that we're working on right now in the lab. We published a preliminary study, which was cut off by the pandemic, unfortunately. So we only were able to do the baseline studies and not the ones where we used repeated stimuli to try to get a chronic effect. We're working on doing that now. But when we looked only at baseline, we saw some proof of principles that looked promising. We were able to see changes in blood pressure and ECGs that were at least consistent with the idea that these participants were experiencing parasympathetic withdrawal and perhaps upregulating norepinephrine. And that was true even when they were not actually pedaling the bike, but were only doing virtual exercise. So we're continuing to work on this. This is an exciting concept to us. It's something that we can discuss more in the Q&A if you like. But I have just a few minutes left, so I'm just going to spend those few minutes telling you about using some of these exercise molecules as therapies for diseases. So one of the diseases we thought would be, one of the family of diseases we thought would be a good test for this are the polyglutamine diseases. So these are diseases that are inherited in humans and have variable lengths of expansions of glutamine-rich regions, which cause aggregation of proteins and cause neurodegeneration. And to do this, we're working together very closely with our collaborator, Sokol Todi, who is an expert on polyglutamine diseases and has made fly models which express the human versions of these disease proteins. And by overexpressing the human version in the fly neurons, the flies begin to have the same kind of neurodegeneration that patients and mice do. Their mobility becomes restricted and their lifespan is reduced. Okay. So we chose 3 different diseases to try because we're aware that from literature about these diseases in mouse models and to a lesser extent in patients, that there are highly variable results. Exercise helps in some studies, in other studies it doesn't. So we wanted to try several and see if there were particular diseases that were more amenable to exercise-based treatments. And what we found was that SCA3 did not respond to exercise very well at all. SCA6 responded a little bit, but not very well. So I'm not going to show you those negative data. I'm just going to show you the one that actually did respond very nicely, which was SCA2. So the black and gray that you see here are ones where the disease gene was not induced, and those improve with exercise as normal. But when you put in the disease gene into these guys, first of all, they go down much more steeply with age without exercise. But if you exercise them, look what happens. They start out going down the same, but at some point, after 3 weeks of exercise, this trajectory changes, and they begin to run not only as fast as wild type unexercised, much faster than diseased unexercised, but they're actually running as fast by the end as the wild type exercise, which is quite extraordinary. And we see the same thing with endurance as well. And you may not be able to fully appreciate just how dramatic this is if you're not used to looking at this particular type of graph. So I'm going to show you just an actual video of what these guys look like when they're running. So to orient you before the video starts, these guys are the wild types. These guys are the ones that have the overexpression of the disease protein, and you'll see that they're able to run not very well at all. And you'll see these guys are running quite nicely like wild type flies. So the key one to look at is the middle here. So these are flies that are expressing the disease protein, but have been exercise trained for 3 weeks. There is no genetic or age difference between these guys and these guys. The only difference is that these guys receive 3 weeks of exercise training. So take a look as you can see they're doing much better than these guys. These guys are barely able to get off the ground. We'll restart. And here you see these guys are actually going up at pretty much the same speed as the regular ones. And if they're not exercised, this is what they look like. So for us, this was really dramatic and really exciting. And so we went on to look at whether this was happening by reducing the formation of aggregates or by some other mechanism. And we found indeed it is. Exercise by itself is capable of reducing the amount of aggregates that are forming in these guys. And if we compare that to ataxin 3, which is not responding to exercise in terms of improving mobility, you can see it's also not reducing the aggregates. So somehow that's the difference. The diseases that are going to respond well to exercise are going to be the ones that do reduce these aggregates when they're exercised. What is the mechanism that makes them different? Well, we have a hypothesis that it has to do with which disease proteins are susceptible to being unraveled through autophagy and which ones need to be unraveled through proteostasis. The ones that can be unraveled through autophagy, we think, are going to respond because cestrin and iditarod are going to be powerful drivers of autophagy downstream of exercise. So we decided to see if we could produce the same effect just by co-expressing cestrin along with the disease protein. And this is what happened. So here you see the mortality of the flies expressing the disease protein. They're having a much higher mortality and dying much sooner than the controls. Now, as I add in the ones that are co-expressing cestrin, we see they got a modest benefit to mortality. It is statistically significantly improved from the ones expressing the disease protein without cestrin. But you can see that it's also not quite bringing them back to wild type. However, when we looked at mobility, we saw something different. Here's the mobility of the controls, which are normal. Here's the mobility in the disease proteins. It's reduced across ages. But when we put cestrin back in, look what happens. That's the green line here. It's now running exactly as fast as the wild types, even though it's expressing the same disease protein as these guys. So this tells us that cestrin is a really good therapy for restoring mobility to animals carrying this disease protein. And it also tells us mechanistically that mobility can be separated from mortality. So if this is a fly, obviously not a patient, But if we could extrapolate to a patient this type of therapy, even if it doesn't completely improve the mortality, it's going to improve quality of life substantially by restoring mobility. And that is also quite valuable. So we're excited about that. Okay, so just to summarize, I talked about a lot of different things, just trying to give an overview, but I hope these are the main points that I've convinced you. Drosophila is a useful model for studying genetic components of exercise adaptation. Octopamine, the invertebrate norepinephrine equivalent, when given in a pulsatile and controlled fashion, can mimic exercise adaptations systemically. And we've identified a highly conserved axis that mediates exercise adaptations in muscle of the fly and is also conserved with vertebrates as well, involving cestrin, PGC1 alpha, and the FNDC5 irisin. We also think that both VR stimulation and overexpression of cestrin are potential therapeutic treatments for patients that cannot exercise. And I showed you data from our recently published paper looking at neurodegenerative polyglutamine diseases. And I'll call your attention to something I didn't talk about, but which is now in press and should be coming out any day now in disease models and mechanisms. We've also used some of these same approaches to rescue a mitochondrial disease in flies called Barth syndrome. So this is not only neurodegenerative, but other diseases that impair mobility as well can be responsive to these kind of therapies. And now I just want to show you the people who did the work. Alison Sikowski did most of the early work on octopamine and cestrin, and she is now in the lab of our collaborator Sokol Todi and has continued to work on SCA polyglutamine diseases and exercise in flies. Tyler Cobb is working on the Iditarod project, and the rest of these lab members are working in other projects that we're very excited about that we hope to be able to tell you about in the months and years to come. And these are our collaborators at University of Michigan. Junhee Lee collaborates with us on cestrin and Iditarod, and Miriam Greenberg collaborates with us on Barth syndrome, and these others collaborate with us on projects that I didn't have time to talk about today. And these are our funding sources. And so I can stop there and take some questions.
Thank you so much. This was such a nice talk, and thank you also for sharing your unpublished data. I think everything is really exciting. So yeah, if people have any questions, they can place the questions here in the Ask a Question button, or also in the chat if they will. I can ask few questions while giving time for people to type their questions. I don't know if I missed it. The lysosomal activity that you showed, is that only in muscles or is it in all tissues of the fly?
Which activity is that?
The lysosomal activity exercising?
Yeah, so it goes up the most actually in adipose tissue. And we think that relates to the increase in lipolysis that's coming about during chronic exercise. With exercise itself, we don't see a huge increase of that in muscle and heart. But if we overexpress some of these factors like cestrin and Iditarod, those can drive increased lysosomal activity also in muscle. And we think there is increased activity in muscle. We just think that it's probably a little bit more transitory and so harder to pick up with this assay, whereas the adipose tissue are really cranking it out continuously.
Right. And kind of related to this, have you checked TOR signaling in different tissues after exercise?
Yeah, so we think that's the key way that cestrin is actually working is by downregulating TOR complex 1. So I didn't really talk about it in detail, but if you look at that cestrin paper in Nature Communications, we looked at all of the downstream requirements of cestrin, which are various parts of the TOR complex pathway, and we're able to show which of those were required for the effects of exercise and which were dispensable. And it turns out that what is most important is downregulating translation through 4-EBP and upregulating autophagy, and also simultaneously using TOR complex 2 to upregulate AMPK and AKT. So it's that combination of things going on that really is the characteristic group of things that you need to coordinate these kind of adaptations.
That is so interesting. Well, I have more questions, but now we have questions from the audience. So we'll start with a question from Rita. She's saying, thank you for a fascinating talk. I might have missed it. Do you have any evidence that the VR treatment can induce physiological changes in the fat body, for example, or any other tissue in flies?
Well, we haven't tried the virtual reality on the flies. We've only tried that on the humans. And we didn't look at the human's adipose tissue. All we looked at was some just things we could get from blood samples. We looked for the norepinephrine itself, and we had live ECGs to measure the heart rate variability and things like this. In the fly, however, not through virtual reality, but through octopamine, we have been able to show that there are substantial transcriptional changes in the whole body. We have not done transcriptomics, however, specifically only on adipose tissue. We did it on whole body. So there may be differences in particular tissues that are actually not being picked up by the whole body one if they're localized only to that tissue. So that's something we would like to do in the future. It's a good question.
Thank you. Another question from Miriam. Hi, great talk. It looked like for the neurodegenerative disease, female flies were used, while earlier you stated that females don't have as much dopamine release during exercise. Maybe I missed it, but how do the females still benefit from exercise in the disease model, but not under normal circumstances?
Yeah, that's an excellent question. So the answer is, well, there's two answers. So first of all, they benefit from the exercise itself. We don't exactly know 100% why. So we know that if we take normal wild type ones, they don't get better with exercise, but there are several examples of ones that are impaired where they start to come back to normal with exercise. Okay, so like these neurodegenerative ones have very low endurance and speed, and exercise is able to kind of raise them back up toward wild type. So we don't know exactly why that is. It could be that without octopamine, you have sort of some level of response that you can have, but you can't drive it past the normal wild type. We don't know. But in terms of when we're overexpressing cestrin, that's a different story. So females definitely respond when we're overexpressing these downstream molecules, the same way that they respond if we go ahead and feed them octopamine or activate their octopaminergic neurons. Once we get to the downstream components, they respond to those the same way as males.
Thank you. Another question from Rebecca. Any ideas about how this relates to octopamine inducing male-male aggression?
No, I really don't know. But although, so we have a theory, we have no evidence for this. So I guess I should call it a hypothesis, not a theory. But so one question that we get asked a lot is, why in the world would they evolve such that females don't respond to chronic exercise? And we don't know the answer to that. I can tell you that we have looked at other Drosophila species besides melanogaster, and it's actually particular only to melanogaster that it has this sex difference, which is lucky for us because it allowed us to do these genetics to narrow down the region of interest. But other Drosophila species, you see some where actually the female are better. You see some where they're the same. You see every different combination. And so what we think is that there's simply not a huge amount of selective pressure on the ability to respond to chronic exercise with healthier aging in a short-lived animal like a fly. What we think is that the evolutionary pressure that's driving these different sensory responses to octopamine is probably related to something else, like perhaps the aggression, perhaps even more likely the courtship, since courtship is a kind of a classical hot area that you can intervene as evolution to start speciating is by changing courtship, right? And since octopamine controls that, what we kind of think happens is that evolution divided out melanogaster through changing courtship and aggression, and then the response to chronic exercise just kind of came along for the ride, but was not really what was being selected directly. But
And
we don't have any proof of that at all.
is there any difference between virgin females and mated females, or is it the same?
Yeah, it's a good question. There is a difference. And the virgins tend to be better runners than the mated ones, but they also don't respond. And also, we have tried looking at ones which have mutations that make them sterile, thinking that maybe it was just the diversion of resources to reproduction that was causing the difference. But even sterile mutants, they also don't respond to exercise, and sterile males do. So it seems to be unrelated to reproduction directly.
Mm. And have you tried the same experiment instead of feminizing the males, masculinizing the females?
Yeah, so we did. If we feminized the females as a control, to make thinking, what if transformer itself is having an effect rather than just feminizing? But the feminized females look exactly like normal females.
Mhm. Okay, and there are a couple of more questions, but I think they have been somehow answered already. The first one is, thanks for the talk. Do you have an explanation for why female flies don't improve their performance with exercise, even though they do have the octopamine neurons and receptors necessary?
Yeah, so we think that it has to do with excitability somehow, because they're receiving the same sensory inputs, but the octopaminergic neurons are not depolarizing. So one of my students has that for a project right now. They're marking these neurons with GFP, sorting them through FACS sorting, and then doing RNA-seq on them and looking for genes that are altered only in exercised males, but not in exercised females. And we hope that we have some candidates that have come from that, where she's repeating those experiments to make sure they come out from multiple rounds of RNA-seq and are reproducible. And as soon as we do that, then we'll take each of these candidate genes and actually put them into octopaminergic neurons and see if that restores the ability for females to respond. But the kind of candidates that we're getting out are things that relate to ion channels and things that actually sort of make sense with the idea that it has to do with excitability, so that somehow in the females, when they receive the same stimuli from exercise, it's just not getting over the threshold to depolarize those neurons.
And last question, and again, I think this has been explained, but thank you for this interesting presentation. Was there a specific reason for choosing Drosophila for this research project?
Yeah, well, we were already working on Drosophila before, so we used to. The lab has always been focused on cardiac and skeletal muscle aging, so that's what we're interested in. And we just thought that since exercise plays such a huge role in determining how well muscles continue to perform with age, both skeletal and cardiac, we just thought that the fly system would be a great system to get at some of the complicated genetics, because you can just do things in much larger sample sizes and with much more fancy pants genetics interventions that are kind of expensive to do in mice, where you're breeding together 3 or 4 different types of mice. In flies, it's much cheaper and easier to do these kind of things, and it's easy to get very large sample sizes so that you can really nail down whether things are changing or not. So those are the reasons we wanted to do it in the fly.
Thank you for your answers. I have just a really quick one, maybe crazy. How will it be to feed humans with octopamine?
Yeah, so people have actually tried that. So the human adrenergic receptors can bind octopamine. They bind it with a lower affinity than they bind norepinephrine. But actually, people have proposed this, and you can find octopamine in health food stores if you want to try it. the actual human studies that have tried stimulating with octopamine have not been very promising. It just doesn't seem to do that much, to be honest. But in the fly, where they have no octopamine, and it is, their receptors are evolved to bind it very strongly, it has a much bigger input. I think to get the same kind of response in humans, you would have to go with epinephrine or norepinephrine, which again is not, because we have a closed circulatory system, it's a little bit dangerous to start tampering around with a lot of, things that are going to change your blood pressure. And so maybe that's not so recommended.
Right.
But this is why instead what we want to do is physiologically stimulate the brain to coordinate it in a way that is physiological and does have feedback controls from your own body. By using the virtual reality, it should be much safer.
Yeah, yeah, I really like that. That's super cool. So yeah, thank you so much, everyone in the chat. I don't know if you can see it, but it's thanking you for such a fantastic talk. So really, thank you for being with us today, and thank you also everyone for joining. We are going to have a little break next week, but we are going to keep you updated with our seminars in October. So for now, this is it. I already shared here in the chat the Zoom link for the Q&A session. So everyone is more than welcome to join us. And thank you once more, Robert.
Thank you for inviting me. It's a lot of fun.
See you, everyone. Bye bye.